Hey everyone, Vivian, you just beat me to the punch. Hi everyone, welcome to the review. I hope everyone is feeling fun and funky and fantastic today. Just before we start, I wanted to get into the secret password question. I don't think we've spent enough time talking about the history of genetics in class. And so as a part of the secret password question for this week, I wanted to talk about some history. And let's bring it up really fast. I could bring it up on that one. Yeah. Okay, so the password question for this week is, in which organism did Barbara McClintock and Harriet Crichton first observe crossing over, which was the first time anyone had ever observed crossing over? And the answer to that is corn. So let's talk about it. Got it right here. So a little macroscopic history of genetics. So obviously we had Mendel doing the very most foundational stuff back in the 19th century. And then we had this guy. And then we had some continually nuanced interpretations of genetics and new discoveries, such as this guy, Edmund Wilson, discovering sex chromosomes, Calvin Bridges learning about sex-linked inheritance, and then, of course, the superstars, Barbara McClintock and Harriet Crichton. So genetics, well, I personally like to feature women scientists because they're criminally underrepresented in scientific history. But genetics is actually a field where women have kind of dominated throughout the history of the science, just because genetics was something that a lot of male scientists didn't dabble in. They must have considered it less important at the time. And, it was highly unintuitive, all the genetics that was going on. So women scientists, biologists would find themselves working on genetics. And I think it's a pretty cool science. So Harriet Crichton and Barbara McClintock. Barbara McClintock was an absolute superstar in the study of genetics. Let me get there. She was kind of an eccentric person, and she had a lot of crazy ideas that ended up being perfectly true, the stuff that we study here in class. And this is her, Barbara McClintock. She wanted to become a professor. She was a professor, but a lot of universities didn't allow women to gain tenure at universities. And they said, oh, you're a woman, you can never get tenure. And so she said, okay, well, then I'm not going to be a professor anymore. So she just stuck to research. Ignore the cigarette. They didn't know that smoking cigarettes was highly carcinogenic back then. But yeah, she did. She transitioned from professorship to some very intense genetic research at very highly prestigious institutions. And she had a particularly impactful discovery that she made with her friend Harriet Crichton. And Harriet Crichton was a school teacher, and she wanted to be a researcher, but because she was a woman, her opportunities were limited. So what she would do is at nights, after she had finished up at school for the day, she would go over to her friend Barbara McClintock's lab, and they would do some late night genetics research. And what they did one night in the 50s, they witnessed rather, actual crossing over happen between two chromosomes. So what they noticed was that this extra piece, or there was one chromosome with a knob on the end that had an extra long piece. So, it was very distinct compared to another chromosome. And what they saw was that these two got close together. And what ended up happening is the one that used to have a knob and a long end ended up just having a knob. The long end obviously transferred over to this chromosome via crossing over. This was the first time anyone had ever discovered, or conclusively discovered, crossing over in genetics. And another very, very important aspect of this is what I don't think we've talked about it yet, but a phenomenon called wait, I'm blanking on the term, transposable elements. That's right. And so transposable elements are the idea that, for example, you have this chromosome, let's say it's chromosome 9. Well, actually, yeah, it is chromosome 9. Let's say that a gene right here, for mechanisms or reasons that we still don't understand, using mechanisms that we still don't understand, would just get up, maybe even go to a different chromosome, chromosome 2 or 3. It transposed itself from one chromosome onto another. And whoa, Barbara and Harriet discovered this. And actually, humans are, all organisms are full of transposable elements. They're an essential part of the reason any biological organism functions. And we still, like I said, don't really understand them yet. Humans are about 40% of our genomes are comprised of these transposable elements that we still don't really understand. So, you know, that's a lot of us that we don't really know about. Corn is famous for being more than 90% transposable elements. Their genes are just like getting up and shuffling around and doing a bunch of crazy stuff. So yeah, two trendsetters, absolute foundational geneticists. And they studied corn.
Gosh, that's so cool.
Yeah. And we will actually, unfortunately, see a trend of female scientists being ignored because Barbara and Harriet's discovery was seriously fought against. Like they tried presenting their findings way back in the 50s, and they did it for decades. And all the male scientists were out there shouting them down, saying, "You're crazy, get out of here. You're not allowed to present at our conferences anymore." And then in the 80s, people were starting to think, you know, I think genes get up and move around. And Barbara was like, yep, I found that out back in the 50s, have a whole bunch of data to corroborate it. And then, so she was only in the 1980s given the Nobel Prize of Science for work she'd done back in the 50s. And we'll see that women scientists are often ignored throughout history. A very famous example being Rosalind Franklin, who we'll talk about. Okazaki-sama, who we'll learn about in, what's that unit called? I think it's replication. But yeah. And I think that was all I wanted to say. Does anyone have anything? Yes. Yeah. DNA. Yeah, does anyone have any comments or ideas to add on? All right. Oh yes, Rosalind Franklin was, well, like I said, we'll talk about her, but she discovered the shape of DNA and its structure. But unfortunately, she was not allowed a prize because she died after or before it was discovered that her work was stolen. But yeah, we'll talk about it later. It's a travesty, for sure.
All right. So do you want to get started, Garrett, on going over the questions?
Oh yeah, actually I saw that Casey is here and was wondering if she wanted to talk about her question from the email now or later. Hi. Firstly, sorry, Sam, can you take the host away from me?
Wait, what do you mean? Sorry.
Can you claim host over the meeting? I just needed my one of my accounts to host so I could share those screenshots on my PC, but now I'm
How
switching
do
over.
I make you unhost?
I don't know if it's possible. Earlier, it offered me the chance to take over the host, so I thought someone could, but now it's not working.
I didn't get the chance to look at Casey's emails, but are are they the two screenshots that are attached?
Yes.
Do you
Wait,
want me to
actually, I don't know.
share, or are you able to
Yes.
do it now?
I was able to reclaim the host, so we're all good.
Okay, cool.
But if you wanted to share and talk about it, that would be great.
Yes. Or actually, I have not had the chance to read the email yet. However, Casey, I have made a question that's really similar to the one that we went over in class. So do you maybe want to see how we do that one and see if it answers your question? Because they're like basically identical.
Yeah, that's fine. Whatever is easiest for you guys. I'm sure one way or the other, I'll gain an understanding.
Okay, Garrett, do you want to start going over your first question while I read Casey's email to see what's up?
Absolutely, that sounds like a great idea. Let me pull it up. Am I muted? No, we're good. Let's share our screen. Actually, one second. Okay, sorry, I was muted. So question number 1. We're talking about gene order. Can anyone tell me what this business is that we're looking at? What is this figure, this table? What's it all about? It's okay, no problem. I don't actually think we even talked about it for some reason. This is what we call a 2-point cross. And, did we talk about this in class? I know we did 3-point crosses.
I cannot remember, but this does relate to linkage groups. So
Yeah, okay. So I'd like to ask you, in what order are these genes found? And like Sam said, we can break the genes up into different linkage groups, groups that show like obvious linkage or unlinkage. So where's a good place to start with this?
I'm kind of thinking we would need to see the parental genotype, but maybe not because you have recombination frequency percentage. So I'm thinking the higher the percentage, maybe I don't
Yes, you're
know.
almost there.
Highest, like the highest number of progeny we know is like non-recombinant, but this is not what that is. It's not the same thing.
Yes, you were so close. You were almost there. So you are perfectly right. We need to look at recombination frequencies. And what do we know about recombination frequencies? I wish I'd prepared this. I'll talk about it more later. But when, let's say we have our parental genotype, and that is, green and red, I guess. And so if they cross over or recombine at any point, then we'll have one that's like red and green, like this, like they crossed over right here. So the maximum number of recombination that can occur is 50%. And I wish I had prepared that better. But yes, so we can look at recombination frequency. And what do we know about these? I wish I had not erased. Okay, there we go. So what do we know about individuals that are genotypes that show a recombination frequency of 50%? 50% is the max. And what kind of linkage is that associated with? Yes, independent. Why can't I? What's happening? Yes, they're not linked. They're independent, exactly. So with a recombination frequency of 50%, it means that the genes are far enough away that if we have a gene over here and a gene over here, that when they recombine, this gene will end up here and this gene will end up here. If we had a gene that was here, when it recombines, it's still going to be here because the distance between this green gene and this red gene is just too small. And we know that recombination frequency is kind of like a distance, a map unit, we call it, because if it's close on this genetic map that we see, this depiction of our chromosome, then we know that when it recombines or when it crosses over, a lot more of the or a lot less of the progeny are going to have shown recombination because these two are so close together. However, if they're very far away, like this green and this green, when it crosses over at this point, as much or as many as half of the offspring can have shown this recombination. I don't think I explained that well, but yes, 50% recombination frequency is the maximum number possible. And this high frequency shows a lack of linkage between two genes. So if the distance between A and B is 50%, A and B are unlinked, right? So let's draw our chromosome. I can't draw a straight line. So A and B are totally unlinked. So A is pretty far away from B. We see that A and C are unlinked. So A is pretty far away from C. So maybe we shouldn't even draw A for the moment. We know that A is kind of far away from B with maximum recombination frequency, far away from C, maximum, and far away from D. So A is kind of like in a world of its own. But we see that B is kind of close to C. It's not at the 50. It's not like 1 or 2, which would be really close, but it's still not far away, not too far away. Similarly, it's even closer to D, and C is also not too far away from D. So I'd say B and D are the closest because they show the lowest percentage of recombination. So let's say that B is close to D. And we also see that B is close to C. But C is kind of further away from D than it is to B. The distance between B and C is 20. The distance between D and C is 28. So I'd say C is close to B and a little further away from D than it is to B. So C, B, D. And then A is kind of off doing its own thing. Does that make sense? And we can even write it out to make sure that we've done it right. So C and B are 20 away, 20 map units. B and D are 10 away. I could draw this better. So B and D are the closest. C and B are the next closest. Then the distance between C and D is a bit more. And we can see that there's kind of a discrepancy between the distance between C and B and the distance between B and D as it relates to the distance between C and D. And that's just because as we get further out, we can identify fewer recombinant individuals. And that's just, I forgot why, but that's just a little fault of the science. And then A is just so far away that it's unlinked to the others. So we kind of see two linkage groups. We have A all by itself, and then CBD is a linkage group. And so this is the order that we'd find our genes. And it could be that D is over here. We know that B is in the middle because it's the closest to D and C. So we know that B is in the middle. But this could be DBC. A is just so far away, we can't really relate it to any of the three. But yeah, this could also be this way. And this is still 20. And this is still 10. And this is still 28. How do we feel about that? This was a 2-point cross. And if you want a tougher one, you can find it right here. And if we have time at the end and you want to, we can definitely go over it. Makes sense. Larger recombination frequencies, further alleles are from each other. Exactly. If anyone wants to go over this one later, we definitely can.
Okay, cool. So now we can go over this double crossover question. And Casey, I read your email. I think that we should, or like me going over this, hopefully will answer your question. If anyone has any questions as we're going through this, just type it in the chat or unmute yourself and let me know, because like I know that at first they can be more difficult. And so the first thing that we're going to do is find out what the middle gene is, given like a bunch of data. So you're given all these different genotypes and the number of progeny that have these genotypes, and we have to find the middle gene. So does anybody know where to start? Yes. Okay. So we have one person in chat who said S is the middle gene. And so that's correct. Does anyone want to explain why S is in the middle or how they came to that conclusion?
I can try to explain real quick.
Yeah.
So the reason why S is first off, we gotta look at whatever has the least number of offsprings, I guess you could say, and that would be 11 and 14. And then you compare that to the ones with the highest number. That would be 356 and 472. Wait, 347. And then say you would compare 11 with 356 because you have the W and the X matching, but then you see that the S is not matching, meaning that you had a crossover of the S. And then when you look at 14 and you compare it with 347, X and W are the same as 347, but S is not. So that would say that there was a double crossover in the middle gene that got changed with S.
Yes, that is a perfect explanation. And literally, I don't think it could have been said any better. So does that part make sense to everyone, how you go about finding the middle gene?
So is it S that's in the middle or S plus? Because
It doesn't matter. You could say S or S plus. Most of the time, it's people normally just say S instead of S plus, but both are acceptable answers.
S and S plus are two alleles that represent the same gene. So the same gene is still in the middle. It doesn't really matter which one or which allele you say is in the middle.
okay, that makes sense. Thank you.
All right. And so after we find the middle gene, then we have to find the recombination frequencies. And by recombination frequencies, we mean what's the frequency of recombination between S, or in our case, S and X and S and W, because we want to know how often we're going to recombine between the two genes that sandwich our middle gene. Does that make sense to everyone? All right, I'll take the silence as a yes. So the first, I know that Dr. Chiari said during class to like rewrite all of these so that like S is in the middle, but honestly, like you don't necessarily have to do that. Sometimes I feel like it's kind of a waste of time while you're, yeah, Ori is right. Like it takes a long time to do during the test. So basically what you can do is, let's say first, I can't remember which one I typed first. Okay, we want to find the distance between S and X, and that's the one that we're going to do first. And so the distance between S and X, we have to find everywhere where our S and our X don't look like either of our parentals. And so one trick that's nice to know is that the numbers for your double crossovers are going to show up in both of your recombination frequency calculations. So the distance between S and X is going to include 11 and 14, and the same thing with S and W when we do that calculation. So we know for sure that recombination occurs between S and X for 11 and 14 because, well, there are double crossovers, but if you look at S and you look at X and you compare it to either of our parentals, you can see that they are not the same in either parental. So that means that we had crossing over occur between S and X for 11 and 14. And now we have to figure out between 91 and 74 and 78 and 83 where crossing over occurred between S and X. So if we look at 91 and 74, if we look at S and X, we can see that we have S plus X and S X plus, and neither of those look like our parents because our parents were X plus, S plus, and XS. So that means that crossing over between S and X took place for our 91 and 74. And so that would be the numerator. So the numerator is just the number of progeny where crossing over took place. And then you divide it by the total progeny. And if I unhighlight this, this is what you should get at the end. So you'll have your 11 and 14 from your double crossovers, and your 71, or sorry, 74 and 91 from your single crossovers. And if we look down at the last two lines, the reason why we didn't include 78 and 83 in our calculation for SX because because we have SX and S plus X, which are identical to our parents. So does that make sense so far, how we found the recombination frequency between S and X? Okay, I got one yes. Casey, does that make sense so far? Because I think that's where you were tripped up, right?
Yeah, but I just need to write it all out because like reading it on the screen, it's not making sense. But I think if I wrote it out, it would make sense. But also, why would Dr. Chiari tell us to rewrite all the genotypes with the gene that's supposed to be in the middle? Is that like gonna water it down more? And
No, I think she has you guys do that because it's easier to compare. Because like you'll see when we start comparing S and W, you have to like look from here to here. But like I don't think it's that big of a deal to have to do that. I mean, like you can rewrite it if it makes it easier, like visually for you to see, but it's not necessary.
Yeah.
Okay. So now we can find the frequency between our middle gene and the other one that we haven't done, which was W. And so this is, like I was explaining, just like I just said, this is where maybe like you might want to rewrite all of your genotypes so the S is in the middle. But like I said, I think it's a waste of time, if I'm being honest, especially because you don't have endless amounts of time on the exam. So now we're going to compare S and W. And so again, like I said, our double crossovers are always going to be included in our calculation. So we'll have 11 plus 14 again in our numerator. But now we have to figure out which of our single crossover progeny are going to be included in that calculation where they had crossing over between S and W. And so here we can kind of use process of elimination because there was already crossing over between 91 and 74. So that means that the other one was probably, or that means that 78 and 83 had crossing over between S and W because there are single crossovers, but also we should always double check and look. And you can see that when we're looking at our S and our W, we have S and W plus, which does not look like either of our parents. And we have S plus and W, which also does not look like either of our parents. So that means that we had recombination and crossing over occur in those two individuals between our S and W. And so 11, 14, 83, and 78 are going to be the progeny where recombination happened between S and W. And then again, you divide by your total, and then you'll get this number. And so that's how you find the recombination frequencies between your middle gene and your other two genes. Is that making sense to everyone?
Sam has a little thing at the bottom of the talk that explains it very well.
Oh,
And,
yes.
I am also going to make a video going through several 3-point cross examples so that, because I want you guys to just become like 3-point cross masters. So it's kind of an afterthought and you don't spend a bunch of time struggling on it. So we're going to work through it together in some videos and it'll become second nature to you. And also Sam's steps stepwise guide is very well done.
This is kind of like a cheat sheet if you
Yes, Casey, I do that. So I'll do that in my video examples. And I can
Okay.
also do another one, so you don't have to wait.
Joseph, you have a question?
Yeah, can you hear me right now?
Yeah.
Okay, so like you said between the S and the X alleles, but does that mean like there is crossing over between the S and the X? Is that what you mean by saying between?
Oh, yes.
Okay.
Sorry. All right.
I'll ask a quick question if that's okay before you move on. So
Oh, yeah, of course.
basically you look at like once you figure out the gene that's supposed to be in the middle,
Mhm.
to figure out the recombination frequency, you're looking at the distance between the S being sandwiched, which is like you have written S and X and then S and W. So for each example, you exclude the non-recombinant because that's going to look like our parents and that's what we're comparing to. So we're comparing the double crossover and the single crossovers to the non-recombinants.
Yes, exactly.
And if any of those are different from the non-recombinants, then you include that progeny number in your count.
Yes. So your non-recombinants, which are your biggest numbers, will never be included in your calculations.
Okay.
Oh, just to see if I'm thinking about this right. So the way you determine the middle gene as S is like you look at the one with the lowest number, which is 11 and 14, right?
Uh-huh.
And then you would compare each one of those to the parental one.
Yes.
So would you compare W plus X plus and S, the one with 11
Uh-huh.
individuals? Would you compare that with the 356 one or the 347?
356. So you do it, this part is kind of like, I guess there's no like formula for it, but you would compare 11 to 356 because it differs only at one place. If you try to compare 11 to 347, you can see that it differs in two places and it wouldn't make as much sense.
Gotcha. Okay. Okay. So even though the one with 91 differs in only one place, we would not say that's the middle gene. Why exactly again?
Because it's not our smallest, it's not like the genotype with the smallest progeny, because so basically what happened, the reason why we compare the two genotypes with the smallest number of progeny with the greatest number is because the likelihood that you're going to have crossing over happen twice is very, very, very low compared to the likelihood that you'll have crossing over happen one time. And so the fact that crossing over happening twice has a very low likelihood, that also means that the likelihood that you'll have progeny who have who had crossing over occur twice is low. And so you'll have the least number of those progeny. Does that make sense?
Yeah, okay. That makes sense. Thank you.
Okay, cool. All right. So the only other two things really that we could ask you for double crossovers is about the coefficient of interference and interference, and they kind of go hand in hand. And so basically the coefficient, oh yes, Casey.
Thanks. I didn't want to interrupt, but before we move on, I do have another question in regards to recombination frequency. So I like went through it. I'm just like trying to see the screen really well. So for S and X, like whoa, I don't know what I just did. Okay, for S and X, I see how you have like 11 is included because W plus X plus S is different from the parent. I see 14, I understand 74 and 91. But
Mhm.
looking at 78, W plus XS is also different from the parent, and as well as 83, WX plus S plus. So why are those not included in the SX one?
So the reason why they're not included in SX is because we have to look at S and X on their own. Like we don't, when we're looking at SX, we don't care about the W's. We just want to look at S and X. And so if you look at 78, we can see that S and X, if you look at just these two, are the same as one of our parents. And then if you look at X plus and S plus, we can see that it's also the same as one of our parents. So because between the S and X genes, because they're the same as our parents, that means that no recombination happened between S and X. So we were looking at the recombination of S and X, and so that's why we didn't use 78 and 83 in the first calculation.
Yeah, that totally makes sense. So in the comparison of S and W,
Mhm.
why did we include 83? Because it looks like 83, we have W and S plus. Oh no, I see. I was looking at 91. I was looking at something different. Okay, that's why, because that's different. Yeah, that totally makes sense. I can see how like you can figure this out without rewriting it, because that's very simple.
Yes. I'm glad that it's like starting to make sense now. With these kinds of questions, it does take practice and just like doing a few of them so you can kind of see like the pattern and how it works.
Yeah. So,
Yeah, thank
oh yeah,
you.
go ahead.
Oh, that's all I got. Thank you.
Oh, but going off that point, so when you were finding, like, on Casey's point that why isn't the 78 and 83 included in the SX recombination, right? That recombination frequency. How did you know to compare 78 with 347 instead of like say 78 and 356? I mean 356. Yeah, wouldn't that like technically show like a crossing over?
So the reason why we compared to 347 instead of 356 is because when we're finding the recombination frequencies down here, we only want to look at single crossover events. And so if we compared 78 to 356, that could have been a double crossover event.
Okay, because both the X and the S alleles are changing.
Yes.
Okay.
All right, cool. Does anyone else have any other questions about that? Those were all really good questions.
I just want to make sure, because I had a very similar question. But kind of what I told myself in my head is basically, as long as they match one of the parents, as long as they match one of the non-recombinants, then we're good.
Yes.
Okay, so I don't have to like, I guess, think about double crossover, single crossover, just it's okay to think about as long as they match one of the non-recombinants.
Yep.
All right.
As long as it matches one of the non-recombinants, then it's not a crossover. All right, so we can always come back to this if anyone else has any questions. But now we can talk about the coefficient of interference. And or yes, is the coefficient of interference different from the coefficient of incidence? No, I don't think so. Sorry, I think I just got my eyes confused. And I was just typing it all and I was thinking interference and I wrote coefficient of interference also. That was my bad, guys. Okay, so to find the coefficient of incidence, basically what you do is you take your the number of actual double crossovers that you saw and divide it by the number of expected double crossovers that you'd expect to see. And so we can see that obviously the observed number are going to be our 11 and 14 because that's like from the data. So that's what we observed. And then to find the expected, you multiply your individual recombination frequency. So you multiply your single crossover recombination frequencies by each other, and then also by the total number of progeny. And so what you end up getting is (11 plus 14 over our, so these were our observed progenies, and then, or sorry, observed number of double crossovers, and then we divided it by our two individual frequencies times the total, and that gave us our coefficient of instance. And then, Ori, I see that you have a question and your hand raised, so hit me with it.
Yes. So I do remember that during class, we were finding the distance for the recombination frequency. Wait, am I looking at something different? I am looking at something different. I apologize.
Oh, no, it's all right.
My bad.
No, don't worry. All right. Does fine?
Can we go over that again? I'm trying
Yeah.
to match what you're saying with the slides,
Uh-huh.
but I just didn't follow what you said, really.
Oh, no problem. Okay, so what we do, so coefficient of instance is actual number of double crossovers divided by expected number of double crossovers, right? And so knowing that, obviously, or maybe not obviously, but our actual number of double crossovers is going to be just like straight up taken from the data that we have here. And so at the very beginning of this question, like before we started solving anything, we established that the genotypes with progeny of 11 and 14 were our double crossovers. And so that's where we got 11 and 14 from here. And then in the denominator where we're trying to find the expected number of double crossovers, in order to find the expected, we have to multiply the frequencies of our single crossover. So that was SX and SW, because when you multiply those two individual frequencies together, it gives you the frequency with which you should see double crossovers occurring in your cross. And so we multiply the 0.18 and the 0.176 together, and that gives you the frequency of seeing double crossovers. And then the reason why you have to multiply by the total is because if you did just 0.18 times 0.176, that's just giving you another frequency, or that's giving you like the predicted, or it's giving you the frequency of having a double crossover, but then you have to multiply that by your total to find the actual expected. Is that making sense to everyone? Casey, does that?
Yeah, that makes sense. Will it always be multiplying the two independent or the two single crossovers?
Yes.
Okay. So if there were like 4 genes, 1, 2, 3, 4, then and there would just be another frequency to multiply together.
Yes, but I don't think we'll ever deal with 4 in this class. That's like, it's out of our wheelhouse.
Okay. Okay. Thank you.
Yeah, no problem. Okay, so incidence slash interference is basically like, how was there stuff that occurred while were there things that occurred while we were crossing our two genotypes that would have resulted in a greater number of double crossovers or a smaller number of double crossovers from happening. And so here, I feel like once I talk about the interference, it'll make more sense. And so to find interference, you just take 1 minus our coefficient of interference. And so when you do that, you end up with 1 minus our 0.746, and you'll get 0.254. And so because this is a number greater than 1, that means that we saw less double crossovers happen than we expected. But if it was a number less than 1, then that means we would have had more double crossovers observed than expected. Does that answer your question, Joseph? So it's kind of just like a way to see if we saw more or less double crossovers than we thought we would. All right, cool. So does everyone feel a little bit more comfortable with double crossover questions now? Because I know at first they are very daunting. Okay, Casey says a little bit. Alex says yes. If at any time like you want to go over it again, we can at the end if there's extra time. Also, Garrett said he's going to post those really awesome, great videos, also noted down here. And this is kind of like a step-by-step guide about going over and solving double crossover questions. So hopefully with all of those things, you guys will be able to do them in your sleep with little effort. All right, Garrett, you want to talk about the next question?
Yeah. Okay. Number 3. So aneuploidy, we just talked about it yesterday. Let's talk about it some more. So what enables aneuploidy, or what is one of the primary causes of the different aneuploid conditions? Very nice, simple, one-word answer. Any ideas? Non-disjunction, thank you. Let's see if Sam, can you unhighlight these as I reveal the answer?
Oh, yes, of
Thank you.
course.
Sorry, yeah, my computer's being mean to me. So yes, non-disjunction, which is a failure of either homologous chromosomes or sister chromatids to separate during anaphase. And non-disjunction might be a word that is just like, whoa, that's a long word. It's intimidating me. But it doesn't have to be. And let's break it down. So we have non-disjunction. So junction is when two things are together, they're joined, such as homologous chromosomes. Disjunction is when these things become separated, like when they're pulled apart during anaphase. And non-disjunction is when the disjunction doesn't happen. For example, if we are in anaphase, our guys are being pulled apart, but let's say that they're not pulled apart. They experience non-disjunction. And what should have been, sorry, this pen isn't working the way I want it to. What should have been this, where our two homologous chromosomes separate, instead becomes this. They non-disjuncted. They did not undo their junction. And so we end up with one cell that just doesn't have any DNA. Obviously, that's not viable. it's going to die. And this one has twice as much as it should. And this basic failure to separate is what causes the different aneuploid conditions. I wanted to say something else, but I forgot. Anyway, so what do you think? Is aneuploidy going to be more of a severe threat to organisms if it happens in mitosis or in meiosis? Yes, I did, Joseph. I took your advice. So Casey and Joseph both guess mitosis. And I'm just going to come out and say that that is incorrect. Meiosis is when we see the really impactful, sorry, yeah, it's fine. It maybe not be too intuitive, but hopefully I can explain it well. Yes, the answer is meiosis. And once we think about it, it's actually quite simple. So let's say you have a skin cell, a skin cell. It's a somatic cell. It's going to undergo mitosis. So if your skin cell ends up with a monosomy or trisomy, if one skin cell has this funky genotype, do you really think that's going to harm the organism too much? Not really. But if you have instead a germ cell which undergoes meiosis and it ends up with this trisomy, this is going to go on. This is going to be a gamete that ends up building an entire organism. So this entire organism now has trisomy, and that comes with a whole host of problems, as we've seen in lecture, or maybe as we haven't seen, because Dr. Chiari focused on maybe what doesn't go wrong, but some things that can go right. And that is true. Non-disjunction isn't always bad. But yeah, if it happens in meiosis, you're going to have the offspring's entire genome be this abnormal, possibly very detrimental case. But if it happens in like a skin cell or a muscle cell, it doesn't really matter. So yeah, somatic cells can do whatever, but aneuploidy and meiosis creates gametes with drastically different distributions of genetic information that will contribute significantly to the offspring that receive it. And a follow-up question, we could either do this or not do this, depending on what you guys would like. It is a very important question that I'm surprised we didn't go over in lecture. What zygote genotypes can result from a cross between a normal parent and a parent that shows non-disjunction in meiosis 1 versus in meiosis 2? Thank you, Sam. So to reword that question in a less confusing way, what is the difference between if you have a parent that has non-disjunction, or actually, so let's say you have one parent that undergoes meiosis normally, and they produce this genotype. So normal gamete, sorry, that handwriting was bad. Normal gamete from our normal parent and a gamete that undergoes non-disjunction from a parent. So you start off here, 2N. And so let's investigate what would happen in meiosis, or if non-disjunction happened in meiosis 1. So meiosis 1, we are in anaphase, and our guys don't separate. So this is going to create, can you see this when I'm writing over the text like this? Yes, okay. So we have non-disjunction happen. There's another chat message. Sure, I can change it if it's not doing it. Sorry, I don't always think about the things that I do. Dun, dun, dun. Okay, so another chat message. Okay, sure, no problem. I thought lighter colors would help, but so we have our parent who starts off as 2N, they are going to undergo non-disjunction in, God loves us, cannot write, meiosis 1. So these two that are lined up in their homologous pairs are supposed to split up in anaphase, but they don't. So we get this. Two different cells. This is that end result of meiosis 1. And when we go into meiosis 2, I guess I should underline this. Then when we go into meiosis 2, we have this. Obviously, this will, during anaphase, still end up as this. And this in anaphase will end up as this. Because the two homologous chromosomes will now separate in anaphase of meiosis 2. And so then this is going to get paired up with, sorry, shouldn't have been, that should have been this. So this is the normal gamete that comes from a parent who started off like this at the beginning of meiosis 1, and it ends up like this at the end of meiosis 2. And so this is going to combine with this, and we end up getting an offspring that has 1, 2, 3. This is a, what's this called? I can't even think. Oh, this is trisomy, because we have the normal 1, 2n plus 1, or I guess this would be 1, 2n plus 1. So if non-disjunction happens in meiosis 1, meiosis 1's anaphase sees non-disjunction, then what's going to happen is that someone raised their hand. What's up, Ori?
Quick question. In non-disjunction, sorry, is it always going to be no separation of either of the sister chromatids at all, or it could happen that one of them gets separated but the other one doesn't?
Could you repeat that, please?
So, like, say with non-disjunction in meiosis 1, could it happen that one of the sister chromatids does get separated, but the other one remains together?
Oh, you mean like if this one right here got separated?
Yes.
Became this? Yeah.
Okay, just wanted to double check if that was a possible scenario. Thank you.
Nothing's impossible. I don't think I'd ever, well, when I was in genetics, I didn't have such an example, but yeah, I'm sure that's possible. I'm sure that's a thing that happens. And then we would have, well, I guess we can do that math later. But yeah, we'd end up with an organism that would there be 2 trisomy and 2 haploid? What we'd end up with is 2 trisomic and then 1, because this gamete would still go to reproduce with this one, and it would end up as this, because these original cells ended up with nothing. So this would not be trisomy, this would be monosomy, 2n minus 1. So you'd have 2 monosomic children and 2 trisomic children, or gametes, I guess. That's what would happen if non-disjunction occurred in meiosis 1, where these 2 homologous chromosomes didn't separate, but instead wound up in one cell. You'd end up with half of the offspring being trisomic, or having trisomic, being the result of trisomic gametes, and then the other half monosomic. What's up, Joseph?
Yeah, so in class we talked about some example where there was like a carrier or something like that.
Mm-hmm.
So would this parent that shows non-disjunction, would that be basically like a carrier of a disease that stems from this non-disjunction?
This parent who experienced the original non-disjunction would not, because this was not a mutation, but a non-disjunction event that happened just by chance in their cell. It's not something, because they didn't start with an abnormal ploidy number, but via their own cellular processes developed one via non-disjunction. This individual, if it were to reproduce, actually, well, this is not viable in most cases, except for on chromosomes 21, 18, 8, and 3, if I remember correctly. But no, what you're thinking of is, actually, we can talk more about that later. That's a great question, but this is not the, would you say like inherited or, I don't remember the specific name, but this is not that. I remember the slide you're talking about. But yeah, this isn't it. However, this was what would happen in meiosis 1 non-disjunction, or if non-disjunction occurred in meiosis 1. However, if we had our original guy, that would end up giving a gamete like this, and that's normal. And then we had non-disjunction occur in meiosis 2. So meiosis 1 goes off without a hitch. You end up with this. Meiosis 1, anaphase, these two properly separate. And then they start to drift apart in telophase, and they drift apart in cytokinesis finally. But then in meiosis 2, if there's a non-disjunction event, let's say that this one goes off normally, and the two sister chromatids are separated from each other. Bing bong. However, in this cell, it does not go off well. The sister chromatids, non-disjunct, they do not remove their junction like they should. And what ends up happening is that you have this and this. And so when this normal gamete comes in to mate with any of them, I'm sure you get the point. I'm going to stop drawing these crazy arrows. So when the normal gamete comes in, the resulting cell is going to have 1, 2, 0, 1, 1, 1, 1, 2, 2. If non-disjunction happens in meiosis 2, then you're going to have one of the 4 offspring as trisomic, one as monosomic. Remember, trisomic is 2n plus 1, and that's what we have, 1, 2n plus this extra. Then monosomic, which is 2n minus 1, because 2n, 1 and 2 minus 1 makes 1. And then we have 2 normal diploids. That's the difference between what happens non-disjunction in the different meioses. What's the plural of that? Anyway, any questions?
Also, at the end of everything, you would have what zygote genotypes? Like,
What's the, well, I guess the genotype.
genotype.
The genotype depends on what, genes were parts of the non-disjunction, but if you wanted to have the numbers, in the case of non-disjunction, let me bring out the pen. So non-disjunction in meiosis 1 creates 50% trisomy among offspring, 50% monosomy. Non-disjunction in meiosis 2 creates 50% normal diploids, 25% trisomy, then 25% monosomy. Did that answer your question? Did I interpret that properly?
Yeah, okay, thanks. Yeah, that's helpful.
No problem. Any other questions? So yeah, aneuploidy can enable and has enabled some of the greatest innovations that have ever happened in biology for us living organisms. And at the same time, it is also capable of entirely interrupting our the building of our bodies. And obviously that doesn't get passed on for long.
All right, cool. Garrett, are you good on that question?
Oh yeah.
Okay. So this next question can be kind of confusing. So we are just gonna I'll walk you guys through it because I feel like that'll be easiest. So the question is telling us Bill and Betty have 2 children with Down syndrome. Bill's brother has Down syndrome and his sister has 2 children with Down syndrome. On the basis of these observations, indicate which of the following statements are most likely correct and which are most likely incorrect. Explain your reasoning. All right, so the first thing that we have to know is that Down syndrome can be a result of 2 different things. We could have, it's literally just because you have trisomy 21, which means you have 47 chromosomes. So we can have trisomy 21, 47 chromosomes. Or we can have what's called a Robertsonian translocation, which means that you have 46 chromosomes and you appear normal, but you still have Down syndrome. And the reason why this happens is because you have an individual who has 45 chromosomes, and you're probably thinking, okay, well, he has 45 chromosomes. Like, shouldn't there be something wrong with him? But the reason why it's okay that that person has 45 chromosomes is because the functional part of their 21st chromosome gets translocated onto another piece of another part of a different chromosome. And so they're like fine and functioning and like a normal human being. And so knowing these 2 things, no, I don't think this has anything to do with gene dosage, Ori. And so knowing that these 2 things, we can try to figure out whether the Down syndrome in Bill and Betty's family is a result of trisomy 21 or Robertsonian translocation. And I should also mention that Robertsonian translocation is known as familial Down syndrome. All right. So knowing all of these things, we can deduce that the Down syndrome running in this family is likely due to Robertsonian translocation. And the reason why we know that is because if you have a high incidence of Down syndrome happening in a family, it's most likely because you are dealing with Robertsonian translocation. Trisomy 21 it's more random, and it isn't something that's like a trend that's seen in a family. And the reason why Robertsonian translocation occurs in families more often is because you have that individual who has 45 chromosomes but is normal, and they produce offspring with someone else who has 46 chromosomes, and their offspring start to get funky, basically. So since we know that this is going to be familial Down syndrome, we can start answering, or we can start figuring out which statements are true and which statements are false. So does anybody want to take a stab and tell me if they think statement A is true or false? False, yes. So it's false because if Bill had 47 chromosomes, he should have Down syndrome, but he is normal. And yeah, so basically because Bill is normal. All right. So does anybody want to tell me what they think? If statement B is, oh, yes. Everyone says also false. So yes, it's false because it doesn't say whether or not Betty is normal. However, it doesn't say Bill is normal either, but we're told that Betty has 2 children with Down syndrome. And so since we deduce that this is a result, that the Down syndrome in this family is familial, that means that his sister having children with Down syndrome means that she had to be normal. So Betty is normal because she doesn't have, or because she had children with Down syndrome, but yeah, because Bill has some number of, he has 46 or 45 chromosomes. And we don't know if Betty is normal or not, or we're not explicitly told whether or not Betty is normal, but they have 2 children with Down syndrome. And so the only way for that to happen is if both of our parents were normal. So Bill and Betty have to be normal. All right. So do we think C is true or false? Not quite
Actually have a
true.
question.
Yeah.
So when you said that for reverse-oriented translocation, you have 46 chromosomes, does that mean that the parent has 46 chromosomes, but they appear normal, and then their children get affected due to them having 46 chromosomes, and the children end up having 47?
So 46 chromosomes means the individual has Down syndrome. 45 chromosomes is normal. Does that answer your question? Sorry.
Yes, it does. Then
Okay.
my answer will be changed to false.
Okay. Yes. So that is correct. Bill and Betty's children don't have 47 chromosomes because we established at the beginning that this is due to familial Down syndrome. So okay, cool. So do we feel like D would be true or false? True, yes. So we would be dealing with a true statement because Bill's sister had 2 kids with Down syndrome. And so because she had 2 kids with Down syndrome, that means that she would have had to have 45 chromosomes. And so Bill's sister would have had 45 chromosomes. Yes, Casey, 45 chromosomes implies that she is a carrier for familial Down syndrome. All right, so Bill has 46 chromosomes, true or false? False, yes. So this is false because Bill had 2 children with Down syndrome with Betty. And so the only way that they could have had a child with Down syndrome is if Bill was like a carrier, like we said. So he would have had 45 chromosomes. All right. So do we think Betty has 45 chromosomes, or do we think that she has not 45 chromosomes? So we have a maybe and a true. So Betty would have 46 chromosomes, and I'll explain this one. False. Betty has 46. So Bill's sister is the one who is a carrier who has 45 chromosomes. The reason why Betty cannot have 45 chromosomes is because, like Ori said, familial Down syndrome runs in Bill's family. And so Bill has 45 chromosomes. And so Bill and Betty did have two kids. So Bill and Betty had two kids, and those two kids both had Down syndrome. And so they had to get Down syndrome, or their Down syndrome is a result of Bill having 46 chromosomes, but not Betty having 45 chromosomes. So I guess let me put this in a more like concise statement. So familial Down syndrome, we established, is inherited through obviously family members. And so we said that Bill has 45 chromosomes because it runs in Bill's family. And so the likelihood or the chances that Betty is also going to have 45 chromosomes is like very, very low. And we already know that Down syndrome is a result of it being like from Bill's side of the family and not Betty.
So what you're saying is this: Bill's side of the family has this familiar Down syndrome, but Betty's side of the family doesn't have anything. It is not affected by this familial Down syndrome at all. It's totally normal.
Yeah, Betty's just normal. Betty is an outsider who married into this family. She's not related to any of them except for these two kids, but Betty is an outsider.
So how would you know that Bill does not have it? How do you know that Bill does not have Down syndrome in this case?
We know that Bill doesn't have Down syndrome because Bill and Betty had two kids with Down syndrome. And so if Bill had Down syndrome, then he probably wouldn't. I don't think the likelihood that he would have had kids would have probably been low. Does that answer your question?
Sort of, kind of. I mean, Garrett says it would have been mentioned in the question. Yeah, I get that. But so you're saying there is like people with Down syndrome don't have children generally? Is that what you said?
No, so I did not explain that part very well. Okay, so basically, the reason why we can assume Bill is normal but has 45 chromosomes and Betty is normal but has 46 is because when they procreate and they make a zygote and their gametes combine, Betty's 46 chromosomes combine with Bill's 45. And if you do the math and stuff, I'm not going to draw it out like Garrett does because I am not as talented as Garrett. But what happens is you end up having when you combine your 46 and your 45, you'll end up with some gametes that have 46 chromosomes that are indicative of having Down syndrome.
Okay. So because Bill has Robertsonian translocation, all of his children, if they have 46 chromosomes, are fated to have Down syndrome. Is that what you're saying?
Yes, I think all of their kids would have Down syndrome.
Okay. So it's impossible that they would not. So it's impossible for them to have children that don't have Down syndrome.
I think so, Garrett. It wouldn't be possible for them to have normal kids, right?
No. And the reason why is the same reason that actually there's a question about that later that's going to answer this question. But if 45 combines with 46, it doesn't, not evenly anyway. And so the one lone chromosome, like it's just gonna not have anything to go with, and it might not have all the instructions that it needs. And by chance, it's probably happened several thousand times in the last 580 million years that animals have been going around. That's how deletion events happen, and that's how organisms lose genes. Yeah, long story short, it's possible. Nothing's impossible. It's
So
probably
it's
just not going to be the
It's
exact normal.
possible for them to have children without Down syndrome. Is that what you're saying?
Yeah.
Okay.
Maybe some stuff happens and the trisomy on that one chromosome just doesn't go and it ends up being normal. Who knows?
Okay.
Nothing's impossible. Anything's possible.
All right. Casey said I would have thought that 46 chromosomes would yield a normal child because Betty is normal. So because we're dealing with Robertsonian translocation at the beginning, like I said, what happens is part of your 21st chromosome is transferred onto another chromosome. And so because that happens, you only have 45 chromosomes. And so when Bill and Betty have children, their children have more chromosomes than what they should because they are also going to have all of the genes from Betty's extra 21st chromosome. Okay, cool. And so this last question or this last statement is Bill's brother has 45 chromosomes. Do we think it's true or false? False, yes. So false. Bill's brother has 46 chromosomes because, like we stated at the beginning, this is familial Down syndrome. And so whenever someone has Down syndrome and it's familial, they are going to have 46 chromosomes. All right. And so this just says that the only correct statement is D. So Robertsonian translocations are definitely kind of on the tricky side, and I would definitely highly recommend reading about that section in the textbook. So yes, Joseph, so if we had individuals with trisomy 21, it wouldn't really apply to a question like this because trisomy 21 is random and does not have like any rhyme or reason as to why it happens or like pattern as to how it happens. All right. So now we can look at different types of mutations that occurred. And these questions are definitely much, much, much more simpler than all of the other stuff that we deal with. And so we are given this chromosome, and then we are given a bunch of different chromosomes, and we're just being asked what kind of mutations occurred. So looking at A, what do we think happened? Yes, U and V were swapped. So what kind of mutation would this be called? Not quite. So translocation occurs between two different chromosomes. So translocations, you always have to have two chromosomes that there's like switching going on between. So yes, it would be an inversion. And can you tell me what kind of inversion it would be? There's paracentric or pericentric. Para, yes. So this would be a paracentric inversion. And yep, Casey, you're right. Pericentric involves the centromere. Yeah, the dot is the centromere. And so this would be a paracentric inversion because like Joseph said, we just swapped the U and the V. So does anybody want to tell me what they think this second one would be, what B would be? I wasn't looking for insertion or deletion. I was looking for another one that starts with D though. Yes, duplication. So the reason why this one is a duplication is because we are repeating UV a second time. So we have STUV, UVWX. Insertion would, an easier way to see insertion is if we obviously inserted a gene, but if we did insertion, it could be something like A, B, C, D, E. And if we put the E there, then that would be insertion. So B would be duplication. What do we think C would be? Yes, pericentric inversion. And so we can see that we swapped the DC and the TS, and that included the centromere. And so a way that I always remembered when I learned this material is that peri has an I, and that I means that we include the centromere. Because I remember I would always get para and peri confused, and then I realized that the I is like a helpful way to remember. All right. What do we think D would be? Yes, a deletion. So this is a deletion because in our original, we have A, B, C, D, S, T, U, V, W, X, but here we skipped the W and are completely missing it. So we must have deleted it from the chromosome. And then this last one, what do we think that this one would be? Yes, it'd be paracentric and a duplication. So what happened is we duplicated our W, and so we have two W's now. And then we also flipped this around. So normally it would have read ST UV W, but we flipped it to read WVU. And so those are, yep. So Joseph, we added a W, so we can see that we added this W here. And then after we added the extra W, we flipped it. And that's where we have the inversion. So those are the kinds of mutations that we have dealing with just like single chromosomes. And like those are examples of it. So does anybody have any questions about that? These ones tend to be simpler than the other stuff. All right, cool. If there are no questions, Garrett, do you want to take over now?
Sure. Okay. Yes. Mutation questions are always very fun. I don't know why, it makes me feel kind of like building Legos. It's just satisfying to put everything together. Okay, so question 6. This is just simple multiple choice. There's only one correct answer. Which scenario displays pseudo-dominance? Exactly, Ori. I'm a curious cat. I like to do this stuff. CC. And that was not me saying yes in Spanish. That is the correct answer. So let's go through these answers. When a human has 2 X sex chromosomes and 1 Y, but the Y still determines the individual to develop as a male, what is that? Klinefelter, yes, exactly. Klinefelter syndrome is when you have a male that has, obviously they're still a male because they're determined to develop as a male by the Y chromosome. And as we remember from Dr. Wiggins' lecture, that Y chromosome kind of instantly stops all the pathways and determines that organism to develop as a male. But a Klinefelter male will have more than just the normal 1 X chromosome. They can have XXY, XXXY, 10 X's and a Y, and they're still Klinefelters. And 9 of those 10 X chromosomes will be deactivated by what mechanism? You know, is called gene dosage. Good job. Yeah, good job. But it's not entirely well known whether or not Klinefelter males benefit from female mosaicism, like we talked about in the last review. Anyway, what's B? Cross between 2 pure breeding individuals where the heterozygous genotype is unviable. What's that an example of? Yes, Joseph, the X's would be packed down into bar bodies. And yes, this is the fatal or lethal allele combination. C, when a deletion mutation occurs that extinguishes the dominant of 2 alleles and the recessive allele becomes overexpressed as a result. That is exactly the definition of pseudodominance. Well, I made it up, so hopefully it's the exact definition. But so let's say we have let's look at this genotype right here. And let's say, actually, no, let's not do that. Sorry. Let's say we have actually that same genotype. And it is instead, it's actually made up of this, where you have all capitals on top, all lowercases on top, and that is an A. Let me rewrite it. So let's say that this is the genotype of an individual, and let's say that this C allele gets deleted. And so then the organism becomes obviously ABD, but it still has this C allele to keep it going. And so we still have capital A dominating lowercase a, capital B dominating lowercase b. But now lowercase c, the recessive, is the only allele that represents the C locus now. So it will obviously be expressed. And then capital D dominates lowercase d. So what happens is this individual is going to have a genotype that corresponds to this new recessive phenotype that we wouldn't previously have seen without this deletion. And so this individual, because they experience this deletion and are now expressive of recessivity, they don't dominate because they aren't actually, like, this lowercase c isn't dominating any other allele. They only partially dominate or pseudo-dominate. They don't really dominate because obviously they don't have anything to dominate. I'm not explaining this well, or as well as I wanted to, but yeah, I'm sure you get the point. And so this recessive phenotype will be expressed more than it should be based on the recessive nature of the allele. And D, when 2 alleles coexist in their dominant state within an organism, what's that? We know it very well. Codominance, exactly. Like the colors on a cat. or like your blood type, or like, I'm not going to get into that. So yeah, good job.
Do you mind going back for just a moment?
Oh, yeah.
I'm writing it down.
Oh yeah, no problem.
I'm almost done.
Oh, take your time, take your time. And just in case this was difficult to visualize, we started with S, T, V, U. My V's and U's look so similar. V, U, W, X. And what happened was that an extra W jumped in, the duplication, and then this became inverted. So at this point, it became inverted. So we now have S, T, the inversion, W, U, V. Oh no, did I mess that up? Yeah. I'm not smart like Sam is, so I'm not going to try to do her questions. Let's move on. Is that good, Casey?
Yes, thank you.
Awesome. Okay, question 7. What is this business called? Obviously, we have human chromosome 4 here and a chimpanzee chromosome 4 here. What happened to make them different? See that we had the centromere here and this orange region here. That's now over here. Someone in the chat says paracentric inversion, exactly. So I personally like doing etymology stuff. So we have paracentric. Centric obviously has to do with the centromere, but peri is Greek meaning all around. So you know the changes happening all around the centromere versus paracentric. Para means beside. So it's happening beside the centromere, excuse me. If we had this gene and this gene, if they exchanged, it would happen beside the centromere. It wouldn't be all around the centromere. So yep, just like Alex said, it's a paracentric inversion. But we hear all the time that humans and chimpanzees are so similar. But if that's the case, how do we look so different? How are our muscles built so differently? How is our diet so different? Why, despite being so genetically similar, are we so different? Evolution or cooler, science. All three are correct. Mutation. So we haven't talked about this yet, but it is something that absolutely has to be articulated, is that genes don't act on their own. This orange gene, obviously chimpanzees are more ancestral or less derived. Actually, I'm not going to get into that. Let's pretend that chimpanzees are the progenitors to humans. Well, they're not. What's really happened is that we had an ancestral species, and then somewhere along the line, there is a split, with humans being on one side, chimpanzees being on the other. So no, neither is ancestral or more derived than the other. They split at this node and share a common ancestor that is ancestral to both of them, just as much to one as the other. So let's always keep our terminology straight. But let's pretend that a chimpanzee is less derived than a human, which is not the case. So we had this orange gene here. This orange gene is the expression of this orange gene in reality is affected by this gene here, and this gene here, and this gene here, in this gene here. Most of an organism's genomes are made up of genes that actually control or regulate other genes, like this orange one. And so these genes over here, which we can find right here and here and here and here, regulate the orange gene. So if this orange gene jumps over here, if it's inverted and comes over here, like we see in humans, these genes cannot as effectively, or perhaps not even at all, regulate the expression of this orange gene. Definitely not in the same way that it used to be. And so if genes are expressed differently, we get things like different bone structure, different energy requirements, different diet, different muscular density, stuff like that. And that's why we're so different despite being so genetically the same. Like all of the genes are there, but what makes the genes do their stuff is different because of things like these inversions. Yeah, genes are now disconnected from the genes that regulated them previously, and so they're expressed differently. Any questions about that? Someone in the chat says, gene interaction is affected by the centromere. Not necessarily. It's just that the further a gene gets away from the genes that previously regulated it, obviously, the less regulated it's going to be, and it's going to be expressed differently. This can happen if the orange gene jumped over here in humans. So yeah, it doesn't have to involve the centromere, sorry. Yeah, I could have been a lot less pedantic about my explanation, apologies. But yeah, any other questions about that? Okay. Now, more chromosome stuff. This is obviously a diagram of different hominid species, humans, chimpanzees, gorillas, and orangutans. What's happened to humans? How are humans different? Someone in the chat says, yep, it's the Robertsonian translocation thingy. Exactly. So Robertsonian translocation, we kind of ignored the chromosome orientation in lecture. And I think it's kind of like building Legos, fun to like understand what's actually happening. So chromosomes have different like orientations. We see this chromosome where a lot of it is like beyond one side of the centromere and only a little bit is beyond the other side. And this is called being acrocentric. Acro is Greek, meaning above. So the centromere is above a lot of the genes on the chromosome. So acrocentric chromosome would be something like bing, bong. And then we can have metacentric, and meta means in the middle, I think. And so we'd have a centromere kind of like evenly dividing the chromosome at the center. And then there is, it starts with a T. Actually, maybe I should have prepared this before I started talking about it. So I'm just going to stop talking about it because I can't finish what I started. Anyway, so like Joseph said, Robertsonian translocation. Robertsonian translocation, as far as I know, occurs between, I keep putting the pen away, even though I shouldn't. Robertsonian translocation occurs between two acrocentric chromosomes. So we see here, acro, the centromere is above a lot of the other and above a lot of the other. So these are going to cross over, let's say, like right here and right here. And when that happens, you get these little nubbins at the end. It's going to cross over so that this connects with this, and this connects with this. So we're going to have the little nubbins and then the long sections. Oops. And this is where the centromere is. And this is what's happened to humans. We had previously, all of this information was on 2 distinct chromosomes, but a Robertsonian translocation caused most of the genes, the part that's below this acrocentric centromere. I lost my train of thought. Anyway, you get the point. You can look at the picture. So most of our genes ended up on a single chromosome instead of being on 2 chromosomes. So when this happened, we went from having 2 distinct chromosomes to having pretty much 1 distinct chromosome, because this chromosome doesn't have almost any information on it. It ended up getting entirely deleted from our genome. And this is why chimpanzees, gorillas, and orangutans have 48 chromosomes. Humans have 46, because this Robertsonian translocation business happened on chromosome 2, which reduced us from 48 to 47, and then it happened again, getting rid of 1 of the little chromosomes, ending us at 46. Someone has a question in the chat. Peanut gets axed. Exactly. This little baby peanut gets axed. Very scientific diction. I like that, Joseph. Yeah. Robertsonian translocation, the majority of 2 chromosomes combined into 1, as we see, and the little pieces left behind from both fall away, and the few genes on them are deleted. So an entire chromosome is lost. And that happened twice in humans to reduce our chromosome number from a diploid 48 to a diploid 46. Oh, wow. Apparently that's why we're no longer as hairy. I didn't know that. Oh, that was a postulation. Okay. Maybe that's why we're no longer hairy. You never know. It was definitely selected for when we became persistence hunters, but that's a story for another time. Awesome. So if we don't have any more questions about that, we can take any other questions. We can do the quiz. We can do that extra example that I had at the beginning, whatever you want.
Do you, Garrett, should we pause the recording and say the secret question one more time?
Oh, yeah, sure.
Need that extremely much for the exam, because I remember when we went over in class, I didn't really get it.
Definitely. You're going to need it.
Basically, what you need to know about it is that, let's say like you're a diploid individual, so you're 2N, and then all of a sudden you undergo some kind of mutation, and then your offspring, instead of being 2N, could be 3N, 4N, 5N, etc.
Mhm.
I have a great example. I meant to do more questions that were like about numerical aneuploid conditions and polyploid conditions.
Mhm.
Like if you have a normal genome of like 52 chromosomes, what would you be if you were trisomic, monosomic, and haploid and stuff like that? And those are very valuable questions. And I can
I
pull one up.
I was actually gonna ask if the polyploidies do have anything to do with, say, the 2N plus 1 or the yes.
Absolutely. Yeah. I can show you a question right now, actually. Let me share my screen.
All right, Garrett, I actually have to get going now. So is it okay with you if
Oh, yeah.
I
Absolutely. And if you guys need to leave, you're obviously welcome to do so. But thank you, Sam. Your questions
Thank
were
you,
amazing as
Sam.
always.
All right, bye guys. See
Bye.
you. Have a good weekend.
You too.
Oh, I forgot to share my screen. Bye, Sam. You're a star. Okay, so question. Bing, bing, bong, 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 bong. Ding, ding, bong. Did I skip it? I think I missed it. Or no? Where were we at? Was it there? No. I definitely should have prepared this beforehand. This was what Joseph was talking about earlier. Yes. Turner syndrome. So we haven't talked about this stuff in class, and I really thought we would. Yeah, Klinefelters, we had the question about that. Turner's is when a female has just a single X, and because they undergo dosage compensation anyway and only have one cell with one X chromosome active at a time, obviously this is still a viable organism. Trisomy X just deactivates 2 X chromosomes. Jacob syndrome, that's interesting, where you have 2 Ys. I wonder how that works out. Klinefelter, polyploidy. Okay, I think the examples are here. But yeah, just to articulate it again. So ploid means body or fold. And that's referring to like the chromosomes, because if you draw it out in the silly little way that I always do, you have, your chromosome, and they're kind of like made up of folds. And so poly means many, and poly, many, just being more than the typical 2N that you see in a lot of animals. So you can be a polyploid triploid, obviously triploid being triple like 3, tetraploid, pentaploid, etc. Autopolyploidy. The distinction between auto and allo polyploidy was not well stated, and we definitely didn't do the questions on it like we should have. So autopolyploidy, let's say that you are a Komodo dragon and you are undergoing parthenogenesis. I wonder, does this sound familiar? You have your gametes. So you have a gamete like this, and there's no male around to provide a complementary gamete. And so you are going to undergo duplication. And in this process, you utilized autopolyploidy to go from a haploid gamete to a diploid gamete. And this makes sense. We can break down the word. So obviously polyploidy is a change in the number of our ploidy. Our gametes should be N, but this one became 2N, doubled its ploidy. And then auto means within oneself. So if you have an autoimmune disorder, then your immune system, so autoimmunity, is when your body, your immune system, attacks itself. Autopolyploidy is when your own genome, just by chance, like obviously it's a cultivated ability in the case of a lot of organisms as a way to still reproduce without a mate. They can change their ploidy all by themselves. Plants do this where they go from tetraploid to hexaploid. And actually that's a very poor example and not even correct, but I'm sure you get the point. Autopolyploidy is when something happens within yourself that changes your ploidy. However, allopolyploidy, allo means different. When you're talking about speciation in biology, you can talk about, what's the word? Oh God, recalling me back to AP Bio. That's right. Allopatry versus sympatry. That was a deep memory that I pulled from. So obviously sim means together or same. Patry means fatherland, like patriotic. Two species that are different but sympatric come from the same place. Two species that are different but come from a different fatherland, they come from different places. Allo means different is what I took a long time to say there. And so allopolyploidy is not when polyploidy happens within an organism to change its ploidy, but when two different organisms combine their disparate ploidies to create a new hybrid ploidy. And I'm not going to use red because red's a stinky color. And I chose the eraser somehow. When they combine, what do green and blue combine to make? Oh my God. Let's do yellow and blue that combine to red or green. I'm tired. So we have this new green allo polyploid. Yes, green and purple make purple. Wait, this is what can be done with species that are in danger of extinction. So animals are what we think of when we think of trying to rescue from extinction, you know. Sorry, I actually can't, like with my tablet, I can't scroll up. So if you say something too long ago, there's no way I can see it. So I'm so sorry if I missed your question. But yeah, so we have this original yellow individual. Let's say they were diploid. And then you have this blue individual, say they were 1, 2, 3, 4 tetraploid. So this was 4N, and this is not from this class. This is from when I was a student in genetics. And I'm friends with a professor, so I get to look at it. So we have this individual who is 2N, and this individual, which was 4N. They combine to create a 6N individual. And this is an example of allopolyploidy. And once again, if we break down the word, we have ploidy, which means folds, or like the amount of genetic information we have. GI for genetic information. Poly meaning many, and allo meaning different. So we got this change in ploidy from a different source, from more than one different source. It didn't happen within ourselves like an autopolyploid would, but an allopolyploid acquires its polyploid nature from two different sources, different like allo. That was a very long-winded way to explain your question. Did that help? Did that even answer your question?
Yeah, it is.
Awesome.
I do have another question based off of what we just went over. And would you say that, what's it called? Give me 1 second. Aneuploids are like, do they fall under the umbrella of polyploidy? Question mark. Just because, you know, they were like the quote unquote accidents that happened during meiosis, but technically speaking, you do end up with you can end up with having different types of ploidy because of it.
Oh no, aneuploidy? Well, no, because the idea behind polyploidy isn't like that you don't have your nice pretty 2N anymore. Obviously that's not the case. So we start with our 2N. That's the concept, like the perfect idea we're going for.
Mhm.
Polyploidy is a change, like an overall change in this number,
Okay.
the number that informs the N. So having a 3N, 27N, but aneuploidy, once again, falling back to etymology, ploidy, we know U means true. So if you're a euploid, you're probably this nice, perfect 2N. You have the true number of ploidy.
Mhm.
An, however, means not. So not having true ploidy is kind of, it's not going as far as changing the amount of information or like the base of our information, but instead is just qualifying or being not the traditionally true ploidy. So having 2N plus 1, 2N minus 1, 2N minus 2. Yeah. But that was a great question.
Thank you. It makes sense now.
Of course, of course. Does anyone have any questions? Do you guys want to go over the quiz? Anything? I don't have a time limit. Which quiz? Quiz 7. Yeah, I don't think we went over quiz 7 because we had the exam. I am Gucci for now. I'm glad you are Gucci.
I think I'm good as well, Garrett. I'm just curious. I sent an email.
Oh yeah.
So I'm being patient, but since you're here, I'm curious if you happen to know when the lecture recording from yesterday would be posted.
I don't know. And Dr. Chiari hosts all of the meetings. So that's like tied to her Zoom account. I don't have access to it. So I am sorry. I don't have the answer. You just have to like keep pestering her about it.
No worries, no worries. I just I will be patient. It's
Absolutely.
not urgent. Thank you.
Oh yeah, no problem. Excuse me. I will email her about it though, because I'm emailing her tonight anyway. And I'm sorry, Ori. I promised that I would send out reminders for the quizzes, but I haven't done it once. So yeah, I'm sorry. But Casey, did you want to? Oh, I should not check my email while it's being recorded. Is this being recorded? Okay, it's being recorded. Yeah, I'm gonna get off of my email really fast. Okay, so Ori is okay, or Ori is on. I appreciate taking responsibility, but I'm here for you and I'm here to help, and I should have been there. I was gonna do something. Oh yeah, look at Casey's email. That's why I was there. Okay. Nar, Australian. Did you want to go over this one, Casey? Actually, yes. I could do that. You asked to like rewrite
Yeah.
the question.
Yeah, we totally can, but can we just take like 2 minutes? I really need to go to the restroom.
Oh, absolutely.
TMI, but I'll be back in
No problem.
Okay.
Thanks, Garrett. Have a good weekend.
Absolutely. Thanks for coming, Ori. Bye-bye.
Bye-bye.
You're a star. Bye,
You
Joseph.
too. Bye.
I think I will also cut the recording now. And when Casey comes back, it's all gonna be good. Unless you guys want to stick around, Vivian, Hadi, and Alec. Hadi Abdelkhalim. 2 chat messages. Oh yes, absolutely. Bye, Alec. Thanks for coming. And the secret question, I think Sam deleted it, but I could be cheeky and just press the undo button. Let's back it up. Back it up. Okay, I don't think that's working, and I'm probably messing something up. So I will just say the question I asked was, so there are 2 really important female geneticists. Their names were Barbara McClintock and Harriet Crichton. And the question was, what organism did they first see crossing over happen in? And these 2 women were the first people to ever see crossing over happen. So this was a big deal. And the answer is they first saw crossing over happen in corn. Absolutely, no problem.
I'm back.
Hello. Okay, so sorry if you can hear stuff going on in the background. The walls of my house are really thin. So let's see here. And I'm, like I said, going to do a video on this and kind of do it my way. Sam's way is definitely good. And oh, that's a lot of yelling. I'm not a fan, but yeah, this is a very good resource. Sam's step-by-step guide. But of course we can do it. So firstly, we can rewrite these. So we have our, actually, before we do that, let's do it my way. So we obviously start with the number of parental individuals that have the parental genotype And the reason why this is the largest number is because, crossing over is like a thing that happens. And just in general, in the universe, it's more likely that something doesn't happen than that something does happen. So it's more likely that something isn't going to cross over than that it is going to cross over. That just kind of makes sense. Like it's more likely that a star won't just spontaneously explode than it is that a star will spontaneously explode. Oh, thank you for coming. And if you have any questions, obviously we are always here to answer them if you want to email us or message us in Discord. But yes, thank you for coming and sticking around all this time. Yes, so more likely that something doesn't happen than that something does happen. And these parental or individuals with the parental genotype, they're more frequent because it's more likely that the parent's genotype is going to stay the same. It's not going to cross over and create any of these recombinant individuals. That's just intuitive. And the reason that the least number of individuals, the least frequent, are the double crossovers is because it's even less likely that something will cross over twice than it is that it does once. That's just an extension of something not happening, something happening once, something happening twice. Makes sense. And when we see double crossovers, what we're looking for is, so we have this parental W plus, X plus, S plus, and then this W, X, and S. And with a double crossover, what we're seeing is, let's say we look at just what a single crossover would happen. So a single crossover, let's say that the crossover happens at this locus between the S and the X. So what's going to happen is it's going to share this genotype. And so this first single crossover, single crossover 1 is going to be W, X, S plus. And then if we look at the, or actually it's going to be W, X, S plus, and then of course, actually I should change my color. W plus, X plus, then it crosses over to S. W plus, X plus, S. And then we have our, that is a whole lot of yelling. Then we have a crossover at this locus. So that's going to be this W, and then it crosses over. So we have our second crossover, single crossover 2, W plus from here, then X and S. We're just following the line. And then we also have crossing over on the other side, and that would be W, X plus S. And so we can see, or actually no, yeah, that's just generally how you find the crossovers. But when we start these problems, obviously we're looking for which ones are the double crossovers. So we can, obviously we identify which individuals are the double crossovers, and we compare them to the parental individuals. So a parental individual is going to be W plus, X plus, S plus. And if we compare W plus, X plus, and S, sorry, I'm debating which colors I should choose. So W plus, X plus, and S. Actually, even before that, we can not decide, and we can just look at them and choose which one is different. So we have these are the parental genotypes. Are you with me so far?
Yes, I'm totally with you. Basically, I think like the bottom line is we're comparing the double crossover to the parentals,
Yes.
which you can also just look at the non-recombinant because they're identical to the parentals.
Yes, exactly. Parental and non-recombinant are synonyms. And so we can compare one of these two And at the moment, I'm choosing to compare this, this genotype. So we can compare this to both parental phenotype or genotypes, excuse me. So we can kind of see that the one that's different from one parental is this S. And we can see that the two that are different from the other parental are these two. And so the one that's in the middle is the one that's only going to have one difference between the parentals. And that's because we're looking at the double crossovers. So what's really happening is that we have this S in the middle. I'm going to switch colors again. So we have this S in the middle, the W and the X. And then we have our, actually, I'm going to maintain a consistency of colors. Sorry. So we have our plus and plus and plus. And so what's happening is that a crossover happens here and here. So this W, I wish there was a way to do that. Oh, there is. So when it crosses over, this W is going to switch. Oops. Hey, get back here. There we go. That it's not working. It's not letting me. Oh my God. Sorry. Eek. There we go. So this W that was originally here crossed over here at this first locus. And then because it's a double crossover, this one went over here. Oh God. Oof, this is not going my way. Eek, please just let me pull it. We just need to be patient. If we're patient, oh God, we can do anything, but apparently not.
It's entertaining to watch.
I am glad. Okay, so we have this double crossover, and this is what we see, is that crossing over at two loci occurs. And what we see is that there's two differences in a double crossover because there's two differences, right? This double crossover differs from one of the parental genotypes at two loci. And that's because this W switched with this W, like we see, and this X switched with this X, like we see. And so the S was left intact because it crosses over at two loci that are on both sides of the middle gene. And so our double crossovers, and it's not on the pen anymore, our double crossovers have this S gene that's different. Just the S gene that's different from a parental. So we have our original parentogenotype, W plus, so all the pluses on a row, except for the one in the middle. And then all of the non-pluses on a row, except for the one in the middle. And so then, now that we know which one is in the middle, we can rewrite all of it. And I personally do that because, it helps me sort it out better. So we have W plus, S plus, X plus. I'm writing much too big for this, sorry.
In the example I sent, are we also rearranging the bottom genotype, which was, because it was I think it's the other image. because there's 2 chromosomes. The bottom chromosome is just 1. Okay, I didn't take a picture of what I'm talking about, so
Okay.
never mind. But it's in the slide on slide number 1. Of course, I sometimes add and delete slides, so my number might not match what you have, but slide 52. Can I share my screen? Is that possible?
Oh yeah, absolutely.
Okay. All right, cool. Because that actually, you answered a lot of my questions already.
And like I said, I'm going to do the video and definitely collect my thoughts better than I'm giving now. And also, I'm going
I'm not
sorry.
able to share.
Let
It says
me
it's disabled.
change your permissions then. Allow to share. Where is that? I'll just make you the co-host. Okay, there you go. And I'm sorry to interrupt. I'm going to cut the recording. So thank you for watching if you're watching.